Gene editing is a scientific technique that allows researchers to change the DNA inside living organisms. DNA is the long molecular code that tells every cell how to grow, function, and behave. Think of it as the instruction manual for life. Gene editing gives scientists the ability to adjust this manual, adding pieces, removing pieces, or changing specific parts. This can influence traits, fix genetic disorders, change how cells behave, and even alter characteristics passed down to future generations. Every organism, humans, animals, plants, and even bacteria, has genes. Genes are sections of DNA that act like biological instructions. They determine things such as eye color in humans, disease resistance in plants, how cells respond to infections, and physical growth and development. When a gene is faulty, missing, or contains errors, it can cause disorders or unwanted traits. Gene editing aims to correct or adjust such issues directly at the DNA level. Imagine the DNA strand as a long string of letters: A, T, C, and G. These letters form instructions, like sentences in a book. Gene editing tools act like a very precise search and replace function. Find a specific sequence of letters, cut or change that sequence, and allow the cell to repair the DNA, incorporating the desired change. Modern gene editing tools are designed to be extremely precise, like using a surgical scalpel, not a hammer. One of the most widely used gene editing systems is CRISPR-Cas9. It became famous because it is accurate, comparatively easy to use, faster, and cheaper than previous technologies. CRISPR works like a pair of molecular scissors guided by a map that tells it where to cut. Once the DNA is cut, the cell's natural repair machinery fixes the break. Scientists can influence how it is repaired so that the targeted gene is changed in a specific way. Before CRISPR, gene editing was possible, but it was slower, more expensive, and less precise. Some diseases are caused by mistakes in DNA, like cystic fibrosis or sickle cell anemia. If scientists can correct these mistakes, they may be able to cure the disease at its source rather than just treat symptoms. Gene editing can help produce crops that grow faster, plants that resist pests without chemical pesticides, foods with better nutrition, and animals that are healthier or more resilient. This can aid global food security and sustainability. Scientists can edit genes in cells or animals to better understand diseases and design new treatments. Research exists into modifying mosquitoes so they cannot spread malaria. Gene editing could also help create immune cells that better recognize and fight illnesses. In the future, gene editing may help repair damaged tissues or organs by correcting the genetic instructions cells use to grow. Gene editing raises important ethical questions. Just because we can change genes doesn't mean we always should. Some concerns include safety, where DNA changes might have unintended side effects that appear later. Fairness, if gene editing becomes expensive, only wealthy people might access its benefits. There is debate about editing traits like height, intelligence, or appearance. Many believe gene editing should only be used to treat diseases, not to enhance normal traits. Some gene edits affect only the treated individual. Others can be inherited by future generations. Editing those genes could permanently alter a family line or even a species. Editing populations in nature must be done carefully to avoid disrupting ecosystems. Because of these concerns, scientists, governments, and ethicists are developing guidelines and regulations to ensure responsible use. Gene editing is actively used in laboratory research, agricultural improvement, and experimental medical treatments. Some gene therapy treatments based on gene editing have already shown success in clinical testing for certain genetic blood disorders. However, many applications are still in experimental stages. Widespread use in humans requires more safety testing and ethical oversight. Challenges still need to be overcome, including ensuring accuracy, avoiding off-target effects, and developing effective delivery methods. Research is ongoing to improve these areas. Gene editing gives scientists the ability to change DNA, the fundamental blueprint of life. It holds huge promise for treating disease, protecting the environment, and improving quality of life. However, it also brings important ethical responsibilities. Used wisely, it could help solve major global challenges. Used carelessly, it could create new ones. Gene editing is like rewriting the instruction manual of life, offering powerful opportunities, but requiring careful, responsible use.